thanks for joining us in this sixth webinar of Geofresco Basics project. This project share information on novel tools, has basic substances for the protection of the plants. We want to replace synthetic fungicides with harder compounds that are as well effective, and HEPO works a lot. I just saw today that they released the standard for the evaluation of downy mildew on vegetables. So since this Geofresco is within HEPO, I saw Eva Matjavic that I give you the floor for a short welcome.
Thank you very much, Gianfranco. Good afternoon to everyone. I would like to very shortly thank you very much for good job are you doing together, and also good effort of Gianfranco who is coordinating this all. And I think that there are some very interesting results obtained already, and there are some other in progress. So I would like to wish you good luck with this new year and with all these good results. Thank you very much.
Thank you, Eva. And I thank also Giovanni Baldissera, that is the Geofresco coordinator, that is very close to us. We are more or less at half of the project, and we are having these monthly webinars. So book your calendar for February 15. We will have next one, even in that with speakers from different areas of the world. And in this webinar, so we already had 5 webinars linked to basic substances or potential basic substances. We started at the beginning telling what are those basic substances, how we can use them, and we are going on with experience of different researchers from different areas joining this network that produce data on the application of basic substance already in the list. Now we have 23 and the list is growing, but there are also potential basic substances, so compounds that have the properties of those basic substances and one day may fit into the list. The project is at around the half, and we have several resources on which you can find the presentation. So we have a Facebook page, we have a YouTube channel where there are all the meetings and so on. So you can just Google Geofresco Basics and find whatever you need if you want further information on the project. So I thank all the partners of the project that are working very hard to provide novelties and news that can be useful for the community. And I'm pleased to start this meeting with Davide Spadarro, that is professor in the University of Torino, that will have a talk titled Use of Essential Oils for the Control of Post-Harvest Disease on Temperate Fruit. Be ready to ask questions for after each presentation. That can be into the chat or can be exposed after the presentation. Thanks to you all. Davide, the floor is yours.
Okay, thank you very much for inviting me to give this presentation today. I should say that it would be much nicer to be face-to-face in maybe in a meeting, but as we know, this is not so easy. So we are continuing at the moment with this environment. Anyway, I will today, as already Gianfranco told, give a presentation about our result on the use of essential oils to control post-harvest diseases. I will now share my presentation. Well, first of all, I thank Gianfranco for being a very good coordinator of this project and trying to put together a lot of different people. And okay, I will now start, if everything is correct. Okay, so my presentation is in the framework of this Geofresco initiative. Eufresco, as you know, it's a network of organizations that fund research projects in the area of plant pathogens and in the phytosanitary area. BASICS is a project that lasts 3 years. It is at half, and it aims to demonstrate the activity of basic substances. In this case, today I will focus on essential oils. In the goals of the BASICS projects, there are mainly we are mainly targeting post-harvest diseases and seed-borne pathogens. In my case, I will focus mainly on post-harvest diseases. Essential oils are present in a lot of botanical families of plants. In particular, there are a lot of essential oils which can be extracted from Mediterranean lamiales. They can be extracted from different parts, so from flowers, leaves, roots, wood, the fruit, seeds, and the bark. So a lot of different parts. Mainly, the extraction is generally an oily liquid, which can be extracted from aromatic plant material, which is rich in essences. Depending on the composition, the essential oil can be up to 2% of the weight of the product from which we are taking. They are very interesting because they can produce a lot of antimicrobial compounds which have an activity against fungal pathogens, but also against bacterial pathogens. Normally, essential oils are not pure, so they are not made just by a single compound, but they are a mixture. The fact that they are mixtures sometimes give also a higher activity, a higher antimicrobial activity, which can be due to the different components. In particular, the components which show the highest antifungal activity are normally monoterpenic phenols. And here you can just see some examples of these compounds. So among the most studied in our group, there is thymol, which comes from thyme and savory. There is also carbacrol, which can come from different lamiaceae, and others. Eugenol, also linalol, which is interesting, coming from basil, for example. Normally, when we buy or we collaborate with a company, the first activity is to try to characterize the essential oils, so to know their composition. And to do this, we generally use a gas chromatography coupled with the mass spectrometer. This is a very good instrument and tool that permits to make the chemotypization of the essential oil. Here I just show an example of two of the essential oils we are working more. One is the savory essential oil, and in this case the major component giving the antimicrobial activity is carbacrol, and the thyme essential oil, in this case thymol, is the main component giving also the antimicrobial activity. So one of the first study that we could perform is some in vitro study to see the antimicrobial activity, and in this case we use the agar diffusion test. I show here just a couple of pictures taken from these simple experiments. In this case we use a Petri dish with PDA, then we spread the conidial suspension of the pathogen we are testing, and also another 10 milliliter of PDA. In the center of this dish, we will put a paper disc with a certain amount of essential oil, and then after a few days, maybe 3 days, we are going to measure the inhibition of the mycelial growth. And here just you can see an example of inhibition by basil essential oil towards Penicillium expansum, or by oregano essential oil against Alternaria alternata. Okay, as I told you before, sometimes it is interesting to see if the components of the essential oil can have some antimicrobial activity. And as you can see in some studies, for example, in thyme essential oil, the major components are thymol and alpha-pinene, but the only one which has antimicrobial activity is thymol, whereas alpha-pinene does not have any antimicrobial activity. Similarly, we can see for the savory essential oil, where carbacrol has the major antimicrobial activity and paracemen does not have it. So it's really important also to see the activity of the different components. How the essential oil generally work? They explicitate their antimicrobial activity generally by breaking the lipid layer of the cell membrane, in particular of the bacteria, but also the lipid layer of the mitochondria for the fungal cells. So in this case, they will impair the respiration. And in this picture, which is coming from some quite new publication, we could see that they can interfere with the membrane integrity. They will leave also some compounds outside the cell. So there is a leakage of DNA, RNA, and also protein. They can also block some pump, for example, and also they can create a dysfunction to the ribosomes. So they have a complex activity inside the cells of both prokaryotes and eukaryotes. But as we could demonstrate some years ago, they can also induce some resistance in the host fruit. So the activity is not only antimicrobial towards the pathogen, but it can also be against the fruit. In this case, we can see by using a gene, which is a marker of induction of resistance. The name of the gene is PR8, and we can see the increase of its expression when we treat the fruit with the thyme essential oil. This is just 6 hours post inoculation. After 24 hours, we can see that there is an increase of the PR8 expression when the fungus is entering, infecting the fruit. But if there is a previous application of thyme essential oil, then we can have an earlier response. And this can be kept also after 48 hours. So it means that the reaction response is quite long. Okay, so this is about the mechanism of action. Now about some slides about the efficacy. About the efficacy, I should say that a lot of the first papers were about in vitro activity, and then also about dipping the fruit in the essential oil suspension, or by applying the essential oil by spraying. This is some activity on apple. In this case, we tested 11 essential oils from Mediterranean plants against different pathogens and on different varieties of apple. Here there are the results of the application by spraying of 1% or 10% essential oil emulsions, and we could see in this case that savory and thyme essential oil were the most effective even at 1% application. We did some similar study also on nectarines. And also on nectarines, we could see that the activity was already quite good against the agents of brown rot, so Monilinia lax, Monilinia fructicola, and also Monilinia fructigena, also at a lower percentage, so at 1%. We could also see that when we increased the percentage of the essential oil, maybe we could obtain a higher efficacy in the reduction of the severity of the lesions, but this could cause also some side effect. In particular, phytotoxicity is an effect which could be found very easily on the fruit. This is some application in wound where we could see that the skin of the fruit was really burnt. So after some years with these trials, we decided to change a bit our strategy and to avoid the phytotoxicity issues, but also to avoid the off flavors. The idea that there were some ideas. So one was to reduce more the concentration of the essential oils applied. The other, to keep a good efficacy, is to integrate this treatment with biocontrol agent and thermotherapy, so in particular hot water dipping. Another strategy which could be tested is the use in pre-harvest, so just in the field, few days before harvesting the fruit. And finally, also the application by fumigation, where you don't have a direct contact between the fruit and the essential oil. So about lowering the concentration of the essential oils, we did not have too many experiences. In that period, we also tested the activity of some water extract, which are soluble in water. In particular, it was a project with Argentina, where we tested the extract coming from different plants original from Argentina, but the activity was not so good. So by decreasing the concentration of the essential oils and still applying by dipping, it was not easy to keep the activity. So another possibility that we tested was the application of two post-harvest treatments, both of them environmentally sustainable, so at low impact. And one was a hot water dip together with essential oils. So first the water treatment and later the application of the essential oils. In this case, the results were quite interesting, and in particular, when there was a hot water treatment followed by essential oil application, we could obtain some efficacy which was much lower compared to the single treatments. So we could also calculate that there was a synergistic effect. Then we also tried some pre-harvest application, in this case by going into the field and applying on the fruit the essential oil emulsion at different concentration. In this case, it was a study done on peaches to control brown rot, and this was at the end the incidence of brown rot, natural incidence after 28 days of storage. What is interesting is that there was a significant reduction compared to the untreated fruit of thyme and silicate in particular, and this was not statistically different from a treatment with a chemical fungicide normally applied. So, coming to this aspect of application by fumigation, fumigation is a good treatment. In this case, to do our test, we use some cabinets where we put inside boxes with the fruit, and below we have some petri dishes with a jellified emulsion of essential oil, which is not in direct contact with the fruit, but helps to release slowly the essential oils during the storage. And this can sometimes also be measured. This is one of the first tests that we did, where we could see that the jellification of the essential oil emulsion was a good possibility to be used. And here I show some results of a study where we applied thyme and savor essential oils by fumigation on the fruit. I believe in these experiments it is interesting to show some effect, because on the left side we could see that the essential oils were quite effective. In this case, the incidence of the rots was not so high, so in the control it was around 25%. But what became really clear is that when you have a very high incidence of the rots, here in the control it was almost 100% at the end of the storage, with the essential oil, you really inhibit monilinia fructicola in this case, but other pathogens can occupy the niche. So in this case, Botrytis cinerea, for example, was able to grow and colonize the fruit. So at the end, the number of rotten fruit was lower, but not so much lower because of the growth of development of gray mold. And by looking this in particular later on some in vitro experiment, by looking at the inhibition of the spore germination, you could see that compared to the untreated, the treatments with essential oils could reduce very much the growth of Monilinia fructicola, but on the other side, when tested against Botrytis cinerea, it was not so effective. So this tells us also that you cannot generalize, and sometimes one essential oil has a specific activity, so it's working maybe against a certain pathogen, but not against another. In this experiment, we wanted to see if our slow release emulsors, slow release of essential oils could still release the active components during the storage of the fruit. And here you can see that the antimicrobial compounds still are growing. This is after 1 day of storage, 14 days, and 28 days of storage. So there is a trend of increase of their concentration during the storage in the chambers. And anyway, we could also see that by considering the component of the volatile compounds present in the storage chambers, really thymol and carbacrol were a very low part of the organic compounds. So there are a lot of byproducts which can be produced also by the fruit which are stored in the chamber. So other positive effects of the application of essential oils by fumigation is the reduction of weight loss in the fruit. And you can see, for example, this is the control that all the 4 treatments were able to reduce statistically the weight loss of the fruit during the storage. And an explanation of this could be due to the fact that also fumigation with essential oils can create a coating around the surface of the fruit by reducing, in this case, the transpiration and respiration rate. There is also a positive effect on the nutritional quality, and in particular, if we consider during the time the storage of the fruit, the level of carotenoids and also of vitamin C was reduced less compared to the control when the fruit were treated with essential oils. So now moving to some more recent experiment. In this case, we have been working on apple by tasting these same essential oils. We bought the oils from a company which is called Pranarom in this case, and first we tested their antimicrobial activity in vitro. Here there are the experiments which were done on against Botrytis cinerea. Here we can see the control at low concentration. A compound which is made 100% by thymol, which is called xedatim, and also basil essential oils were reducing, but not so much, the growth of botrytis. In the other cases, the pathogen was completely inhibited, in particular with thyme and savory. And we also studied the efficacy of these oils by fumigation. So all the other tests were only done by biofumigation. In this case, on some apple variety, which is organic, and it is also resistant to apple scab. So it is quite promising for the market of apple. We wanted to see if it was possible to apply essential oils also on this apple variety. And here you can see the results on the quality of the fruit. Really, there was no significant difference in this case, so it did not affect negatively the quality of the fruit. Looking at the incidence of the rots, in this case, Botrytis cinerea was applied on the fruit, and we measured the incidence of the rots. This is the inoculated control. At 30 days of storage in blue, there was quite a very low incidence of rots. At 60 days, this incidence was growing, and it reached a very high level after shelf life. So it was very stress conditional for the fruit. We could see that the chemical control was for sure very good in reducing Botrytis cinerea, but we could see a good efficacy until the end of storage, also for savory and thyme essential oils. So besides this, we wanted to see what is the effect of essential oil application on the microbiome of the fruit. In this case, we sampled the microbiota present on the apple surface, so as epiphytes and also as endophytes, so inside the fruit. And we sequenced an ITS region, and then we performed a lot of bioinformatic analysis that I will briefly show. So about the effect of the tissue, we could see that in general there was these are parameters that measure the diversity and the richness of the microbial population. We can see that the epiphytic population, as expected, was more diverse and richer compared to the endophytic population. Another aspect which we could see is that at harvest there is a higher richness in microbial species, which is decreasing during the storage and then in shelf life. And looking at the treatments, there was not any statistically significant difference between the control fruit, the thiabendazole treated fruit, and the thyme essential oil treated fruit. More or less, we can say that similar results can also be shown by looking at the beta diversity. The epiphytic population is more homogeneous, while the endophytic one in red is a bit more diverse, but they are very well separated. And yes, about the composition, also we could see that at harvest, the sample were really more homogeneous. After storage, depending on the treatment, there was a shift, a change in the microbial composition. Okay, and this is the results of the abundance of the different genera. I think it's quite difficult to be interpreted, but just this is the population of the fruit at harvest, and this is after storage. When we apply Botrytis cinerea, is indicated by this blue, more or less blue color, and we could see that the thyme essential oil was reducing Botrytis cinerea, but thiabendazole was able to eliminate completely this pathogen. We could also see, and this is quite interesting, that after shelf life, the place which was occupied by Botrytis cinerea is later occupied by Penicillium expansum. So also in this case, the niche which is left free by Botrytis can be occupied by a different pathogen. And looking at the endophyte composition, we could more or less see similar results. What is different is that in the endophytic population, there is a lot of components which have not been very well characterized. So, for example, there is a high presence of oculimacula, which is a particular genus. And, for example, Botrytis cinerea can also be inhibited inside the fruit by the essential oil treatment. So I don't know if I am almost at the end, but I will present the last results of a project which is funded by PRIMA. PRIMA is this organization that funds projects aiming to favor the growth of the Mediterranean agriculture. In this case, we have a project which is again coordinated by Gianfranco, and the name is Stop Med Waste. And inside this project, we have two targets. So we work with strawberry and nectarine. And we are also testing here the essential oils which are applied by biofumigation. In this case, we are working with another company which is producing organic essential oils, and its name is Flora. And we started 1 year and a half, more or less, ago, the project. We are testing some essential oils, maybe a bit also different from the previously tested. Well, I should say that the first trial was not so interesting. We just found that some essential oils were increasing the number of the rods, so it was really not so interesting. So for the second trial, we eliminated these essential oils, and we could see that on the strawberries, after 9 days of storage plus 2 days of shelf life, this is the healthy control, and then there are fruits treated by biofumigation with 3 different essential oils. There was a significant reduction of the rods. The fruit were not inoculated because Botrytis was naturally present on the fruit. we can also see that these essential oils did not modify the quality of the fruit. So we would like to continue with these essential oils for next year, for the continuation of the project. And similarly on nectarines. On nectarines also we tasted some different essential oils coming from red thyme, oregano, lemon, fennel, and basil. After 14 days, we could see that some essential oils were quite good, the ones from fennel, basil, and lemon. We measured most of the rots were caused by monilinia on the nectarines. And in the second trial, also we kept these 3 essential oils. After 28 days of storage, the 3 of them were significantly different from the healthy control. Also in this case, we did not inoculate the fruit, but after shelf life, we could see that the activity of the essential oils is really lost. I should say that when you apply essential oils by biofumigation, they have an activity in the storage chamber, but when you open the chamber, you will lose the activity. And here it's we don't have effect on the quality of the fruit. So I'm coming to my last slide, which is about the conclusion. Conclusion is that dipping and spraying with essential oils were our first studies where we understood the mechanism of action of the oil. Later, by different side effects, so phytotoxicity and also off flavors, by doing some tasting of the fruit, we decided to move to fumigation. By using this slow release system, we believe you can avoid to modify the organoleptic quality of the fruit. There are some very good effects on the quality and also on the reduction of rots. And with this, we will continue, and we hope that in the future some of these products could have a practical application. And this is my acknowledgements of the people which have been working on this project, in particular in the last time we are working with Giada Schiavone and Marco Garello on strawberry and nectarines. And I thank also the Eufresco for this initiative and also the Stop Med Waste for funding our research. So thank you very much, and I'm ready if there is time for questions.
Thank you, Davide, for the very interesting talk, and I'm very surprised. So the different talks attracted a lot of people. We are over 100 people that are following us, some of them even on Facebook, and several others. Maybe we'll see the video later in YouTube channel. I saw that there are several questions already in the chat, and if somebody else wants to tell something, can raise their hands. Let's start from the chat. First question is about Royal Gala, and attendant knows that has a short storage because it contains a lot of sugar, and which basic substances can or which compounds can be useful according to you on Royal Gala to be effective to control decay?
Okay, I could say in general on apple, what I would say is that for stone fruit control and also for strawberry, I believe essential oils can have a very good activity, and I would suggest to continue in their in the continuation of their development. I believe in the case of apple, it's probably not the best to apply essential oils, because I believe that well, the rot incidence is quite low. Some prevention and also agronomic practice and good practice at the time of harvest could help a lot. Well, some products are not very easy to be applied, and the companies generally do not like so much the post-harvest application. Anyway, also by doing some experiment in storage chamber with a commercial company, we could see that really it was possible to obtain a good reduction of the rots. So it's a possibility, but I know that the market, the strategy does not like so much this application.
Thank you, Davide. And another question, and you already partially answered with your results, tells that we should move toward the practical application and likely toward the commercial products. This is one of the possible results or achievement or dissemination of this project. So we have to share the knowledge about this, and maybe companies can take interest and go toward the practical application. But the question is to you, your opinion about.
Well, in my opinion, there should be some company which is willing to apply essential oils to a market which is a bit different from the usual, because normally we are working with companies which produce products for medicine or pharmaceutical uses. So in several cases, these companies never thought about enlarging the possible application of their product. So I believe it's also a question of knowledge. Well, during the registration process, there could be some issues which are related to the health of the consumer, but I believe that this aspect could be bypassed, in particular when you are applying biofumigation, because in that case there are no residues on the fruit, and so just by opening the chambers you can leave the essential oils evaporate in the air, and in few hours you don't have the smell also.
Thank you. And just a contribution. There are already several products on the market based on essential oils, with orange, for example, or lemon, or there are some terpenes that are compounds that they showed that are combined together and they are available on the market. So research move toward that direction, and a list of products is available. Another question is if there are compounds that can be used for the powdery mildew.
Yes, some of the essential oils can also be used for the control of powdery mildew and also downy mildew. For example, some, well, not the product that we have tested, but there are some extracts or essential oils with commercial application. In general, not for fungal control. So, for example, there is the essential oil of mint, which is registered, for example, but it's normally used for inhibition of the germination of potatoes and onions. So it has a different application. But for example, the grapefruit seed extract, it's registered for antifungal activity.
I agree, and if I remember well, one of the compounds available on the market in EU based on orange essential oil is registered for some powdery mildews. The problem is that it may kill. It may have phytotoxic activity. So this is a problem that can be discovered in post-harvest, so we need to take care to the concentration, but even in the field, because one of the limit, as Davide told, of essential oils is that a low concentration, or when they are no more, they are in effect at a higher concentration that can be phytotoxic. One more question is about the cost of those essential oil. Is expensive? How much it costs? Is feasible according to you?
Yes. About this, we did an economic study a few years ago, in particular for nectarine and peach treatment, and the cost of the treatment when we use a concentration of 1%, which was anyway quite effective, was an increase of the final cost of 2 to 5 cents per kilo. So I believe it is a cost, but in case you have a high incidence of brown rot, for example, this can be afforded to reduce the losses.
Excellent. A lot of people are appreciating your presentation. You did a very good job. Last, I don't know if it is easy to answer. Can the approximate price of the preparation of such product possibly be related to the surface, weight, or other quantifiable parameters?
It's not clear, the question.
I think it's not easy to answer to this.
Yes.
Okay. Thank you, David.
May I explain?
Sure. Welcome.
Yeah.
The price for the ton, for the quantity which is involved in treatment. Let's say in a storage facilities, there are 2 tons of oranges.
Yes.
How much does it cost to treat? And especially for the efficacy.
Yes.
The efficacy is like a pesticide or not? I suppose less than.
Yes. So about the efficacy, it's less compared to the chemical fungicides. In more or less our experience, it was lower than thiabendazole for pond fruit, lower than fludeoxonil for stone fruit, etc. So it was lower, but in a situation where we are going to eliminate some of these products from the market or reducing their application, or there is also a request from the consumer to have products with a lower level of residues, then it could become an alternative. About the cost of the treatment, that was our calculation of 2 to 5 euro cent per kilogram. So this cost calculated by tons would become 200 euros per ton, for example. So I believe it is a cost which influence in part on the final cost of the fruit. It depends if the fruit has a sufficient amount of earning to be treated. Yes.
Thank you, David. And David claimed for next opportunity to meet together. And one of the opportunities is the Post-Harvest Pathology Congress that is planned in Cyprus at the end of May. And I see here on my screen a lot of people that will be there taking their results. Most of them were already speakers in the previous session. So in case you are interested to know more, and David will be one of the invited speakers, it will be a sub-midway session, and Nikos Sorsakis is doing an excellent job with the other people of the team, organizing committee, and scientific committee. So people that work in the control post-harvest disease and want to know novelties are welcome to digit in Google post-harvest pathology, and they will find the hint. And abstract, it is possible to submit till the end of February. So this is a good opportunity for you that want to learn or to show what you are doing. But let's move to the other side of the world. So I see here Dandan Xu that is ready to share with us her results. So Dandan Xu is from Hainan University in China, and she will have a talk titled Polyphenols Application on the Control of Post-Harvest Disease of Lychee and Table Grapes. Dandan, the floor is yours.
Okay. Hey, Gianfranco, can you hear me?
Yes.
Okay. Thank you very much for giving me this chance to show my research. I'm Dandan Xu. I come from China. My topic is polyphenols application on the control of post-harvest disease of leaves and tip grapes. Polyphenols is a natural compounds found in many fruits, vegetables, and herbs such as blueberries, apples, coffee, and so on. Since polyphenols have higher antioxidants and could protect human against the heart disease, it's a benefit to our human health. So it's widely used in our daily life. Yearly, polyphenols have three major groups. Filler onions accounted for 16%, and personalic guides accounted for 13%. The others are slim beans. As shown in this figure, we can it's the phenylpropionate metabolism pathway. Polyphenols produced from this pathway. In many papers, this pathway have been reported that involved with many disease resistance. Just as listed in these papers, the induced pathway have been related to the disease resistance increasing. In our previous study, we have found that fungicide have induced the delay the disease development of green mold. Just in this figure, this is the control treatment, and the other take grapes are treated with the fungicide, and the disease severity have been reduced. Also, the fungicide didn't inhibit the growth of the pathogen botulism cinerea. From the analysis of gene expressing of PLC4H STS, these 6 genes have been reported to involve with the phenylpropionate pathway. And from the results, we can find that the 6 genes have been highly increased. Also, the accumulation of polymeric compounds have been induced after treatment, after treating with the fungicide oxide. So we guess whether the application of polyphenols could be used on the control of post-harvest disease. We searched the papers and we found that many polyphenols have been used in many post-harvest disease control, such as on the apple grapes, apple polyphenols, and the tea polyphenols, and orange polyphenols. They have been reported on the control of post-harvest disease of sweet cherry, tomato, and apple, and so on. And our research have focused on 2 major diseases. One is the tea grape disease. It's green mold disease, which caused by the botulism cinerea. The other one is leech disease. Leech is a very important and special fruit in China, and its pathogen is Polyphenols leech. And our research, our results have been published in the Journal of PBT. from the we tested the antifungal activity of polyphenols on the inhibition of botulism cinerea, and we found that the tested 17 polyphenols could inhibit the growth of pesticides, and the pterostrobin showed the highest activity. Also, on the other pesticides, pterostrobin showed the highest inhibition activity against the Spongia generation and the mycelia growth. From the SEM observations, the hyphae and the Spongia of these two pathogens have been damaged by the application of polyphenols. This is the results of TM observations. Also, we found that the plasma membrane and the cell wall of these two pathogens have been disrupted by the application of polyphenols, just as in this here and here. In addition, the fluorescence staining of these two pathogens, we found that the plasma membrane of these two pathogens have been damaged. This is the results of in vivo analysis. Pterostrobin should could delay the disease development of green mold, as this listed in this figure. This is the results of this downy blight disease. Also, these compounds could delay the could inhibit the disease development. In cases, pterostrobin showed the highest activity against the pathogen growth. Also, it could delay the development of post-harvest disease. Finally, please allow me to introduce my department. Now I'm worked in Hainan University. This is a map of China, and our university located in here. It's the south province in China. Hainan University was founded in 1954, and it's a key university under the 211 project. So, welcome everyone to China. Welcome everyone to Hainan University. Thanks very much. Please, sorry for my point, please. Sorry.
Franco, turn on the volume.
Muted, yes.
Yes, I am. Okay. Yeah, thanks for your very interesting results, and thanks for showing us your very nice university. And who knows that hopefully soon, one day, we can get there with a big bunch of people to know about what you are doing. And your results were very interesting, and this pterostrobin and other polyphenols as fulvic acid are very promising and can be candidate basic substances, so they can enrich It needs somebody
Yes.
that is ready to produce the dossier and, as it was written earlier, submit to the official institutions, then go through approval. That takes some time, but one of the good side, as Eva told us time ago in one of the previous webinars, is that they do not need a renewal. So once done this work, this lasts for a long time. I saw that there is one question in the chat. This is linked to biofumigation. So at the end, we can have a general discussion. So if Davide Svedari is still with us, we can give him this question. So Dan, which are, according to you, the good side and the bad side of the Peridostilbene or fulvic acid? ETCs, according to you, go toward a commercial compounds. Which are the limitations, if there are limitations for the use toward humans or so?
Sorry, Professor.
Which are the good sides and which are the potential negative aspects linked to the use of those polyphenols as styrostilbene and fulvic acid?
I just found that the pterostrobin have the good have the best activity against among the tested polyphenols. In the future, maybe another and more compounds can be found.
Okay. There is a question: is fulvic acid have phagocytose action?
No. It couldn't. It didn't show the inhibition against the pathogen.
Okay. So according to you, it should be relatively easy to formulate it in a commercial compound. Okay, pretty good. There is any other question or somebody that wants to raise a hand and propose something to Dan? Congratulations to your very interesting presentation, and this can open the way to discovery of new compounds. So very good job.
Thank you.
In one of the next webinars, I see Xiaomeng on the bottom of my screen, so we will learn also from her experience about those compounds. Okay. So thanks to Danda for having the general discussion that we can have. The head speaker is Laura Orsali from CREA DC in Italy, that will have a talk titled "The Seed Treatment in Crop Protection, Essential Oils and Chitosan." Laura, the floor is yours.
Okay. So good afternoon to everybody. I'm Laura Orsali from CREA Center for Plant Pathology and Certification. And the presentation that I'm going to show you is about the seed treatment in crop protection with essential oils and chitosan.
Laura, if you can take the full screen. Okay, perfect.
No.
That's okay. Go on.
No. That was what I had to do.
Impostazione di visualizzazione.
Sì, esatto. Duplica presentazione. Okay. Sorry. Okay. Seed is an essential input for crop production. Okay, seed is an essential input for crop production. Almost the 90% of the world's food crop are grown from seed, and so sowing healthy seed is essential to improve crop yields and increase the food production. Seed treatments represent indeed a crucial and important step for reduction of seed infections. In this presentation, I want to talk to you first about the essential oils. As we already shown, the essential oils are mixture of natural compounds synthesized by plants, and they play an interesting role as alternative strategies to the use of synthetic fungicide for seed treatment, thanks to their antimicrobial activity. Since 2009, our research group has participated at green national and international projects to find alternative strategies for seed treatment against the fungal seed-borne pathogens, with a particular focus on essential oils and chitosan, both excellent candidates as basic substances. As I will show you soon, we have studied essential oils pure or in oil-based formulation, oil alone or in combination with the chitosan for seed coating, screening different essential oils, and then during the years focusing on few oils chosen for their efficacy and applicability. Then chitosan low molecular weight was also studied as an elicitor in plant resistance. This presentation was meant to be a summary of these years of work, and so I'm going to present in terms of in vitro trials, in vivo trials, and then I will show you 3 study cases. The very first activities were carried out to screen the in vitro efficacy of different essential oils against some of the main seed-borne fungal pathogens of the crop here listed. The pathogens were inoculated on agar medium containing 5 different concentrations of essential oil, and then the mycelial growth was calculated. All the oil tested, thyme, tea tree, clove, laurel, rosemary, mint, and oregano, had a clear reducing effect on fungal growth. And that reduction was dose dependent and is different depending on the fungal species. Through the Mickey index, that is the minimum concentration required to produce an 80% reduction of mycelial growth if compared with the growth of the oil-free. So through this Mickey index, we found that the activity of the oils varied depending on the chemical composition. The 4 oil here highlighted, thyme, clove, mint, and oregano, have a higher amount of phenolic ingredients, and that corresponded to a lower MIC and so a higher antifungal activity. But how can we use essential oil for seed treatments? We tested different essential oils. We tested different time of application, starting from immersion in an aqueous solution. But as we know, the oils are insoluble in water, so they need the use of an emulsifier, and so we chose the new film P, that is pinolan-based formulation with adhesivant properties. It forms a film including the oil. Then the use of oil-based formulation could be a means to solve the problem of water insolubility of the oil. Then we test also a spray application with the oil alone or together with the chitosan for seed coating, as we will see in details. Following the in vitro trials, we have focused the activities on 4 oils, thyme, tea tree, clove, and mint oils. The first goal was to assess the best concentration with the higher efficacy and the lower phytotoxicity. In fact, EOS could be toxic to the embryo and reduce the rate of germinability. Studying the toxicity of the listed crop, we found that the toxicity could be affected by different variabilities. First of all, the concentration, because when it increases, the concentration, when the concentration increases, the germinability reduces. Then the crop, because every seed possesses different properties. And so for each couple, crop and tea, we define a toxicity threshold that is a threshold above all the concentration can be toxic and the germinability could be reduced. Then another important aspect is the timing of the exposure. In this case, the toxicity was evaluated by seed immersion. And for all the next trials, we will refer to 10 minutes immersion because it was found to be a good compromise between the efficacy and the toxicity. In the case of chitosan, no phytotoxicity was reported. About chitosan, we made this experiment using an high molecular weight chitosan as a seed coating together with essential oils, with the aim to improve the persistence of the essential oils on the surface of the seeds and so their efficacy against the pathogens. We have investigated the formation of multiple layers on the surface. These pictures are from the scanning electron microscope of the CNR, and through the study of this experiment, through these pictures, it was possible to observe on durum wheat kernel the uniformity and the stability of the structure of the coating with multiple layers, even after imbibition. That is what normally occurs after we sow a seed. And so after the due consideration, the 3 layers were then chosen for all the next trials. Going ahead, the essential oil antifungal activity was tested in vivo on seed, first by seed submersion at different oil concentration after fungal inoculation. The pictures here are from the blotter test method, and the upper showed the wheat seeds inoculated with Fusarium graminearum, where the mycelium is clearly visible. And the picture below, we have the seed of durum wheat inoculated with Fusarium graminearum, but treated with tea tree oil, and we see that the mycelium is reduced. The essential oil treatment caused a significant reduction of seed infection evaluated with this blotter test method. In some cases, this reduction was comparable to our chemical control based on copper. We choose copper as chemical control because copper is the chemical allowed in organic farming. Similar tests were performed on other crops. Pea on tomato, rice, rocket, soybean, and wheat, where we have tested also the spray treatment and another treatment comprising of 2 steps. A first step of immersion and a second step by spray with a mixture of oil and chitosan. Concerning the results, the treatment by immersion, we can see highlighted in red, gave in general better results with an efficacy comparable among the different oil that we test. Clove, thyme, tea tree, and mint oil. The spray treatment with pinolen that we see in blue gave mixed results, varying among the different crops, while the two-step treatment, here lighted in green, gave results more or less comparable with the immersion treatment. I want to capture your attention on the first graph about on the pea seeds, where we see that the immersion with the oil, in clove oil and tea tree oil, gave results more promising than the spray treatment. So please fix this information, and we will see it after. Moving to greenhouse trials, this is an interesting case study on rice. The disease bacane, caused by Fusarium fuscicuroi, causes an abnormal lengthening of the stems and the leaves. So we have higher plants, as we can see in the middle of the picture, while on the right we have healthy plants. The same seeds infected with Fusarium fuscicuroi, but then treated with the essential oil. In the case of the same seeds infected and then treated, we see that the number of infected plants are reduced. We can see in the left of the picture here. So there is a good reduction of the number of abnormal plants. Before going ahead with the case studies in field, I want to introduce the chitosan as a lister in plant resistance. In our work, a low molecular sorry, I can't read. A low molecular weight commercial chitosan was studied as a lister of systemic acquired resistance. And in this work, we wanted to evaluate the chitosan-induced elicitation responses by the changes of some enzymes activities involved in plant resistance and by the change of the phenolic content. After the seed treatment, the small seedling were collected and then studied. And we see that, with the only exception of ascorbate peroxidase, the chitosan treatment, here we see in the middle column, induced an increase of enzyme activity or in a phenolic compound respect to the not treated and not inoculated plants. That increase, as we see in the first column, is what happens in the presence of the pathogen. So when we have the infection after the chitosan treatment, we see a higher response that gives to the plants a better protection against that disease. This chitosan treatment was tested in a field trial on durum wheat inoculated with Fusarium graminearum, and together with other treatment with essential oils. So in this case, both by immersion and spray. So in this case, the chitosan was used both as the lysator and as a seed coating. And in this experiment, we calculated the number of the established plants and the disease incident. Here we have the picture of results of the immersion treatment with chitosan thyme oil and tea tree oil. And we see that all the immersion treatments showed a significant decrease of the number of infected plants with respect to the control not inoculated but not treated. This picture. Spray treatments that here in this slide are not shown because we have really many data. I don't want you to bore more. Gave good results if the oil were applied together with the chitosan for the seed coating. The last case study I want to show you is a 2 years experiment on pea seeds inoculated with fungi belonging to the Ascophyta blight fungi complex. We tested two oils, tea tree oil and clove oil, both applied by immersion and by spray. And the number of established plants was recorded. So do you remember the graph I wanted you to fix? Well, here we have the opposite situation, because in this case, the best results in terms of number of established plants were obtained not with the immersion treatment, but as expected, but with the spray treatment. This is because an excessive imbibition during the seed submersion may have somehow damaged the other seed coats that in the case of legumes are very soft and sensible, affecting so the proper germination of the embryo under the field condition. So concluding, the use of healthy seed is generally recommended as the primary management strategy for the generation of healthy crops and for good harvest yields. The essential oil tested show an in vitro antifungal activity against different fungal seed-borne pathogens, and this activity was dose-dependent and variable depending on the chemical composition of the oil. Do you remember I said that we found that the oil with a higher amount of phenolic content have an higher resulted to have an higher activity. The essential oil tested in vivo reduced on the seed infection, but a great attention must be given to the phytotoxicity. The immersion treatment gave good results in lab and in fields for cereal and for vegetables as well, but not for legumes. In fact, in the case of legumes, it caused mechanical damages to the seed teguments and reduced the germination rate. So for this type of seeds, the best treatment could be the spray application of the oil together with the pineal N as a filming agent. However, further investigations are needed, especially in field experiments, to verify the effectiveness of the treatments in field condition and to assess any possible problem on seeds. Concerning the activity to be shared within the Eufresco Basic project, we have currently a work in progress about wheat kernels treated with clove oil and clove oil based formulation against the pathogen Tillexia levis. And our will is to introduce the chitosan for the next year alone or as a seed coating with essential oils. So I arrived at my last slide. Here I summarize some of the main papers published in the context of seed treatment with all the data that we have collected during these years. So that's all, and I thank you for the attention.
Congratulations, Laura, for the very interesting presentation, and all the people approved and liked all the three talks, and thanks also to Luca Riccioni that cooperated to this project as an Italian responsible for Eufresco. So there are some questions for you. You showed a very long list of results, so a lot of questions can come. Some already are there, others can come, so you are welcome to introduce into the chat or propose directly. The first is about the kind of chitosan. If you use the same formulation, you talked about a formulation that for some people is known. You use several kinds, or it is the same? If you can tell more about.
So yeah, I talk about two types of chitosan. One chitosan of low molecular weight and one with high molecular weight. The low molecular weight we used for elicitation was a commercial solution that is soluble in water. That is the chitoplant. We use that commercial solution. For the seed coating, no, it is not soluble in water, and we use for the spray, we use the chitosan in acid, lactic acid or acetic acid.
The chitosan that is from Sigma or other.
Yeah.
Okay. Very good. Another question is, I guess it is about the production of essential oil. That tells that if there is a large mass of vegetation, can give a reduced amount of essential oil. Are you familiar with those issues?
Well, maybe I don't understand the point of the question, but I have to underline that we are talking about the seed treatment, so not with the mass, not with in field or mass vegetative. So we have to talk about the number of seeds.
Yeah.
So
just a very quick answer for the small what I know is that usually if you have a higher amount of vegetation of the plant producing essential oil, you can have a reduced percentage of essential oil, because essential oil are secondary metabolites. So plants stressed produce a higher amount of essential oil. But if you have a higher mass of vegetation, the amount of essential oil is for sure higher. So it depends on how you manage the crop.
Okay, I didn't understand the question, because I was thinking for the use of essential oil on big field in field with a lot of vegetative mass. So thank you for your answer.
Don't worry, it was just a quick answer to the people that produced the questions. Another question is essential oil utilization must be melanged with adjuvant? This is an important question. How you mix essential oil to be then applicable with which compounds or so?
So we use the new film P, the Pinolan. First, we start using the twin that is a normal detergent used, but we wanted to use the essential oil with a compound that is allowed in organic farming, so something that has an applicability in field. So we use this Pinolan that have a film property, and it is an adhesivant and also an emulsifier, so it can improve the solubility in water, both for immersion or for the seed spraying.
Very good. There is Marisa Tello from Madrid that congratulates with you. Hi, Marisa, for the very good presentation. And
thank you.
As also for a list of other attendants, and we are very happy with that. Another question. Do you have to keep the inoculated seeds at a low temperature, and at which temperature and for how long?
Okay, we used to inoculate, and then after very few days, hours or days, we do the experiment. So we don't have long-term storage. But if we have to wait for some days, we just put in the climatic cell at 5 degrees or something like that.
Thank you. And there is somebody that follows us from Argentina and suggest us to have further webinars. Thanks. We will have monthly ones, so you will be plenty of opportunity. And if you want to see the previous one, you can go on YouTube channel or on Facebook page, and you will find all of them. And even more, there is a research page, ResearchGate page, where all the presentation, and I'm confident also of the speakers of this talk, will be available, and they are also the one available from the previous talk, so you can get them. Feel free to to add further questions. There were a couple for Davide about the use of essential oils. And about you, Davide, to make applicable the essential oils, use of adjuvant, which is your experience?
Okay, yes, about our experience, it's really hard to use essential oils when you want to mix them, in particular with water, so to create an emulsion. So it's necessary, in our experience, to add always a surfactant that can help the mixture and the creation of the emulsion. So I think this is the most relevant aspect.
Okay, and another question is, if you have other questions for Laura, Dandan, and Davide, you can include it to the chat. Another question is, what is the best period to spray the apples, according to you?
Yes, in our experience, the best application that we have tested is the fumigation in post-harvest. In this case, just after harvesting the fruits, and apples, depending on the varieties, are harvesting between August and October. In the experience that we did in pre-harvest, application in pre-harvest, in that case, normally we tested 2 application time just before harvesting. One, it was 2 weeks before harvesting, the second 3 days before harvesting.
Perfect. And another question that I give to you, but it is true also for the other 2 speakers, and I will hear their experience about how long it takes the effect of this biofumigation with essential oils, according to you.
Sorry, how long?
How long it takes the effect of these essential oils?
Okay, it's really short. I would say that it lasts until you keep the fruit in a closed environment. When you open the chamber, you will lose in few hours the efficacy. So as you could see in our experience on strawberry, also on peaches and apples, when you open the chambers, the rods will become the same as in the control. So we should say that, in our opinion, it's more a fungistatic activity and not really an antifungal activity. So it's just the inhibition of growth of the pathogen.
Thank you, Davide. Dandan, are you with us? Okay, in the meantime, I pose the questions to Laura. How long it takes the effect of the treatment with essential oil in your system, and if the application of chitosan changed this or not? You can turn on the voice.
Okay. Well, the application of chitosan, that's the reason why we tested the seed chitosan, to improve the efficacy of the oil, and we saw that the chitosan enhanced the not only the activity but also the effectiveness during the years for storage. So if we have some seeds that we have long storage, that we need to storage for long period before sowing, so it could be an interesting strategy to keep the efficacy longer. And the first question was about the, I don't remember.
The formulation?
The chitosan? Oh, I don't remember. I don't remember more.
Okay, if it come back, we can be back to you.
Okay.
Dandan, are you with us? In the meantime, that Dandan reach us, further question to Davide from Luis Palou that wonder about the organic essential. I think they are biodynamic, but Davide.
Yes, you're right. No, so yes, maybe it makes a confusion, semantic confusion, because the essential oils comes from organic and contain organic compounds. But in this case, I mean that the essential oils we used are coming from a company that produced them in biological farming, in organic farming. And as you said, some of them are certified from Demeter, so they are biodynamic also.
Okay, very good. I saw that there is a, if there are no any questions, I saw that it was a wide appreciation for the 3 talks. So congratulations to all the speakers and thank to the whole attendants that joined us this webinar. And this time we proposed the webinars 2 days earlier and we are happy to have 100 people. Next time, you know that 15th of February, it will be next one. So if you follow our social media, you can get informed about the link for connection. So thanks you all and see you next time.